Hi, thank you for that lovely introduction, and thank you all so much for organizing such an awesome symposium. My name's Eleanore Ritter. I recently started a postdoc at the University of Georgia with Dr. Andrea Swigart, but today I'll actually be talking about some of my PhD work that I did at Michigan State University on the role of clonal reproduction in shaping DNA methylation patterns across flowering plants. So, asexual reproduction, of which clonal reproduction is one form, produces genetically identical offspring. And while we can model this as like a blue square producing two identical blue squares, I think that is a little dull when we have a great Star Wars reference, right? So this is the logic behind Jango Fett's genome being used to produce all of the clone troopers from Star Wars, right? They're genetically identical to their progenitor, Jango Fett. And so this enables reproduction without mates, which, as you can imagine, and as I think we've all kind of, well, not all of us, but as some of us have probably heard throughout the day, is occasionally really necessary, right, when you're doing range expansion and things like that. But this does cause an increased accumulation of deleterious mutations because you're not able to purge them through sexual reproduction. And so obviously that's a pretty large negative genetic effect. There's also suppressed recombination and increased heterozygosity within the genomes of organisms that reproduce asexually generally. Yet in spite of this, asexual reproduction is prevalent across a lot of eukaryotes. So here are just a handful of organisms that are able to reproduce asexually. And what's really interesting is that they're so abundant and that they've evolved the ability to reproduce asexually so many times, despite the fact that it has so many deleterious impacts on the genome. And flowering plants are no stranger to having an abundant amount of asexual species. So clonal plants are plants that are able to reproduce asexually through clonal propagation of basically a vegetative part of a plant breaking off and producing a new individual. And about 66% of Central European angiosperms produce a clonal growth organ. So in theory, they can reproduce clonally. And we don't necessarily have a number for North America or other continents, but in theory, it would be somewhat similar. And so all four of these species are clonal species native to Michigan. And clonality has evolved many times through many different modes. So all four of these actually utilize different developmental pathways and different organs in order to clonally reproduce, some of which are derived from shoot tissue, which I'm sure no one in the back can see that tiny little green icon, but the first three can reproduce through organs that are derived from stem tissue, whereas the fourth plant on the right reproduces through root suckers, which are formed through root tissue. So clonality in flowering plants is very abundant, very diverse, and has evolved through many different mechanisms, despite the fact that genetically, right, we would consider them, quote unquote, inferior to their sexual counterparts. And so what a lot of studies have allowed us to get a better handle on is that the theory that, clonal reproduction, other forms of asexual reproduction produce a lot of deleterious mutations that accumulate over time is in fact correct. Generally, we see a lot of deleterious mutations present in clonal plant genomes. And what we're starting to get a better understanding for, and what this symposium has kind of been about, is the role of the epigenome in modifying other things, including the genome, right? There's kind of this intertwined evolutionary history between the genome and the epigenome in plants, primarily DNA methylation. And so one thing that I think is been very underexplored is if there's any relationship between reproduction and mating systems and the epigenome. And so today I'll be talking about cytosine methylation, which we've talked about a lot today, but I just want to make sure we're all on the same page. So it's a modification made to cytosine where a methyl group is added, and in plants it regulates a lot of really crucial processes like transposable element expression. So when you have a TE or a transposable element, if it's methylated, it generally is silenced and is not able to transpose throughout the genome. However, if it's unmethylated, it's a lot easier for it to transpose throughout the genome, causing genome sizes to balloon and things like that. In plants, gene expression is also generally regulated by DNA methylation, as well as overall genome stability. However, there is a negative impact to DNA methylation, which is why, plants and animals, we're not just coding our genome in methyl groups. One big issue being that cytosine deamination is a pretty common mutation. It's actually the most common mutation to occur in plants, and that's when a methylated cytosine transits to a thymine. And in plants, we generally study these as 3 different contexts, which you might have heard throughout the day. So 2 of which are symmetrical, CG and CHG methylation, where a cytosine is methylated on both strands, as well as CHH methylation, where the cytosine is methylated on just one strand. And they do very different things, which is why we talk about them differently in plants. So this is just one example. So CHG and CHH methylation, when present on a gene body, are generally associated with low expression, whereas when CG methylation is present on a gene body, it's generally associated with moderate to high expression. And so we can't just lump them all as cytosines and call it a day. We analyze these 3 contexts differently. And what's really exciting is that DNA methylation is actually reprogrammed during sexual reproduction in plants. So this is just during pollen production, but all other reproductive tissues and embryos undergo very different reprogramming as well, where ultimately the DNA methylation is reestablished in the embryos. And so in theory, if you're foregoing sexual reproduction and you're not getting this constant reestablishment of DNA methylation, are you just losing it over time? And what we see, at least in CHH methylation, is that maybe clonal plants have lower CHH methylation, basically. So our clonal plants are in blue on this graph with the non-clonal plants in purple. And you can see that the clonal blue species generally have lower CHH methylation. It was unclear at this point if that was just due to phylogenetic relatedness or similarities between these species, or if it was actually due to clonality. And so a big question of mine for this project was to investigate if clonality has an impact on DNA methylation in flowering plants. And so to do this, I used public data of 39 species with a mix of species that reproduce sexually and a mix of species that reproduce clonally, most of which were domesticated. I think there were like 2 wild species, so a very, cultivated plant-heavy data set. And most of them were derived from the stem tissue. As I mentioned before, clonal plants can either develop through the shoot apical meristem or the root apical meristem, and these are all plants that are ultimately producing offspring through the shoot apical meristem. And so to analyze the methylation data, I utilized methylpi, and then I tested if clonality was associated with differences in DNA methylation across the phylogeny using phylo-LM. And what we found, and I'm sorry that's not on a phylogeny, but you can't see the pattern if it's on a phylogeny. Ultimately, if you look, our sexually reproducing species are in dark green and our clonal reproducing species are in light green, and you'll notice that the clonally reproducing species are towards the top of this graph because they generally have lower DNA methylation in all 3 contexts, not just CHH, which actually makes a lot more sense from a mechanistic standpoint than what that previous study found. That being said, this is just a correlation. It doesn't necessarily tell us anything about, the directionality of this relationship. And so one thing that I was really interested in is if transitions in reproductive modes seem to influence the rate of DNA methylation level evolution. And what I mean by that is when we think of rates of trait evolution, if we have a trait rapidly evolving, as you see in pink, right, it's just changing, in this case, body size rapidly over evolutionary time. It doesn't necessarily mean that the body size is becoming bigger. And if we have a slow rate of trait evolution, that trait is not changing a lot over evolutionary time. And so in theory, if clonality does directly lead to differences in DNA methylation levels between clonal plants and sexual plants, what we would expect to see is that basically if we have a species split where a one species is now suddenly able to reproduce clonally, we would see a rapid change in the DNA methylation levels if it truly is getting reduced every generation that it's foregoing sexual reproduction. And so what it would look like would be something like this. And if we utilize a program called EvoRates to actually model this, we would expect something like this, where that whole branch would now suddenly look like it has a really fast rate of DNA methylation level evolution as one species evolves the ability to reproduce clonally. And so using EvoRates, I was able to look at all three contexts again. And this is very tiny, but in general, CG methylation levels evolve pretty slowly with a few exceptions, which excitingly are actually clonal species. So at the Oryza-Rhophopogon and Oryza-Sativa split, with Oryza-Sativa being rice and Oryza-Rhophopogon being a clonal relative, we see an unusually fast rate of CG methylation level evolution. And we also see this in Fragaria ananaza and Fragaria vesca, which are two clonal strawberries. When we look at CHG methylation level evolution, we see a very similar pattern, except the Fragarias are pretty normal. And this time we see Psilocecus heterocycla, or bamboo, is having an unusually fast rate, most likely due to the fact that it is a very large genome, however. And what I thought was really interesting, because I just feel like CHH is always like the weird context, at least in plants, is that it has a really fast rate of DNA methylation level evolution with pretty much no patterns between clonality and sexuality in these plants. However, we do see some clade-specific patterns where grasses up top have unusually slow rates of CHH level evolution, and then asterids on bottom have very fast rates of CHH methylation level evolution. And so this is kind of just the start of this work. So I've actually sequenced a lot more, and so I'm hoping to kind of expand this data set and have more statistical power. But I believe at least initially we can kind of say that clonality is truly correlated with lower DNA methylation levels in flowering plants, at least in this domesticated data set that we have. And this could have really important implications for genome evolution, such as increased TE movement and decreased cytosine deamination. But what I'm really interested in, and why I have sequenced 100 plants at the end of my PhD like an idiot, is that I'm wondering if this is true in wild clonal plants as well as those derived from root tissue, because I didn't mention this, but the shoot apocomerostem and the root apocomerostem actually do very different things in terms of DNA methylation, and so in theory we would see a difference there as well. And so with that, I would just like to thank my co-authors, especially Diego de Sousa, a phenomenal REU student that I mentored a few years back, and my former advisor, my former lab, and Michigan State in the Department of Plant Biology for funding. I'll take any questions. So unfortunately, we did treat it as a binary, and it definitely is more of a gradient, and that's something that I personally would love to have. And there are a few different clades where we actually have data on, like how many rhizomes are being produced. Like Asclepius is a really good system for that. So if I could choose where my future work goes, that would be what I would want to look into. Yeah.
Thank you so much.
Thank you.
Hi everyone, thank you so much for coming. So I'm Arun, and I'm a postdoc at the Technical University of Munich. I would first just like to thank the organizers for this wonderful symposium. It's great to be in a room full of people thinking about both evolution and epigenetics simultaneously. I've actually found it quite hard personally to interact with this group, so it's great to see all of you here. So before I get started today, I'm just going to briefly introduce two major goals within population genomics that's relevant for the talk today. So first is that we're interested in understanding how genetic variation is partitioned both within and between populations. And a second key goal is to reconstruct the demographic history of populations. Now, this has quite important implications in various fields such as conservation, health and medicine, biogeography, and many others. And for the most part, a lot of the work today is really focused on genetic variation. But I'm quite interested in epigenetic variation, and this has probably been defined quite a lot today, but I'll go over it anyway. So I'm particularly interested in plant DNA cytosine methylation. So this is a reversible chemical modification where there's an addition or removal of methyl groups on cytosines. And the reason I'm interested in epimutations is that they've got some interesting properties that makes them different compared to mutations. So with mutations, you can have 4 states. Can I use this? Yeah. I don't think you can see that. Okay. So with mutations, you can have 4 states, corresponding to the 4 nucleotide bases, whereas with epimutations, you can either have the unmethylated or methylated state. Mutations can occur anywhere in the genome, whereas epimutations in plants are restricted to 3 sequence contexts. Mutations not really that sensitive to environmental fluctuations, but epimutations can be. Germline mutations are inherited. And we know that CG epimutations in some cases are also inherited. So I'll actually be focusing on this class of mutations for my talk today. Mutations accumulate at a rate of about 10 to the power of -9 in the model plant Arabidopsis thaliana. However, epimutations change much faster at a rate of about 10 to the power of -4. And this property in particular makes it quite interesting because it means that it might be able to reveal very recent evolutionary events that cannot be detected using mutations alone. However, this environmental sensitivity poses sort of an issue. So what a previous study did is they grew genetically identical sets of Arabidopsis thaliana under various environmental conditions, so things like drought and saline conditions, and they identified regions in the genome where the rate of methylation divergence is not associated with the environment they're grown in. So they've termed these regions clock-like regions. However, this is quite a lot of painstaking experimental work, which is not really feasible for most plant species. And so they also identified a second set of regions that these are the gene body methylated regions. So these are much easier to define. So they are just basically gene exons that have very high levels of CG methylation, but very low levels of CHH and CHG methylation. And they too show this property of environmental insensitivity. So the authors suggest that we could use both these clock-like regions or the gene body methylated regions to understand molecular divergence in methylation. So with this background in mind, we had 2 major goals. So first was to understand how plant methylomes evolve. And the second is to assess the utility of using methylation as a marker for population genetics. So for the most part, we were focused on the Arabidopsis thaliana data set. So I was using the genomes and methylomes generated by the 1001 Genomes Consortium. So we focused on just 2 populations, the Central European one and the Iberian one. And we had about 63 paired genomes and methylomes in each case. So before going into the results, I'll just get some definitions out of the way. I'll be talking a lot about SMPs, or single methylation polymorphism. So these are basically CG sites where some samples have the methylated state and other samples have the unmethylated state. And I'll also be talking about differentially methylated regions. So these are entire regions where some samples have very high levels of methylation and others have very low or no methylation at all. So I'll be outlining 4 sets of results today. And the first one is the insights that we can gather from constructing the methylation site frequency spectra. So the site frequency spectra is a pretty commonly used tool in population genetics because it can reveal how mutation rates, demographic changes, as well as natural selection X within populations. So here's the methylation site frequency spectra, and the x-axis represents the proportion of unmethylated alleles, and the y-axis represents the proportion of sites. So here, a laser pointer would have been helpful, but anyway, so if the site falls into the right part of the spectrum, that would mean that for that site, most samples have the unmethylated state, and if you have a site that falls into the left part of that spectrum, most samples have the methylated state. So here we've generated the methylation SFS using SMPs, or individual CG sites. And we've done it for different regions in the genome. So the top 2 on the left, that's the clock-like and the gene body methylated regions, but also generated it for other regions in the genome. But the crucial sort of thing I want to point out here is that the shape of the SFS is actually pretty similar regardless of where the SMP site was actually, where it was actually taken from. In contrast, If we did the same thing using the DMR states, you see there's now a lot more variability in the shape of the SFS, depending upon the region that it was sampled from. Now, why is this relevant? Partly because there's a lot of interest in characterizing DMR states. You can do it even at shallower sequencing depths, but at least our work indicates that there's some benefit to characterizing individual sites rather than just focusing on entire regions. So the next is the insights that we got from estimating Tajimas D and pi for genetic and methylation variance. So Tajimas D is another very commonly used metric within population genetics, where when you've got very low values below 0, that would indicate forces such as directional selection or population expansions, whereas when you've got very large values above 0, that would indicate forces such as balancing selection or population contractions. So on the panel on the right, that is the estimate for Tajimas D that we generated using SNP. So we estimated Tajimas D across individual intervals, and then on the x-axis you can see that we've classified them depending upon the region that they were sampled from. So the right panel is the estimate that we generated using SNPs, and in the left panel is the estimate that we generated using SMPs. And so when you look at SNPs, or the genetic variants, you can see that the Iberian population actually has a higher Tajimas D value compared to the Central European one, whereas this pattern is flipped when you look at the methylation variant. So here, the Iberian population actually is lower Tajimas D compared to the Central European one. So this is already starting to indicate that at least there is some evidence that evolutionary forces seem to be acting on the two sources of variation somewhat separately. And further supporting this idea is that if we actually simply did a pairwise correlation, so of Tajimas D across that interval for genetic variants, that's on the x-axis, and for the methylation variants, that's on the y-axis, you see basically there's a complete absence of any local correlation whatsoever. And we also estimated pi, which is sort of an estimate of molecular diversity, and again on the x-axis is the estimate based on SNPs, and the y-axis is the estimate based on SMPs, and again you see a very weak correlation. So you might be thinking, well, hang on, how could this be the case? We know that methylation occurs on specific nucleotide bases. However, one possible explanation is that the rates are so different, right? It's 5 orders of magnitude different, so it's quite possible that the evolutionary forces simply are acting at completely different scales and on the two sources of variation. So the third set of results I want to introduce is our efforts to reconstruct the demographic history using genetic and methylation variants. So for this part, I worked very closely with Kevin Kaufman, who is a postdoc in the group. So Kevin developed this deep learning method for inferring the demographic history. So I'll just very briefly outline how his method works. So first, basically, this is kind of he estimates all the genealogies across the genome. So in this case, the coalescence time, or the time to the most recent common ancestor, is directly proportional to the number of segregating sites in that interval. So we estimate the genealogies and then get all the transitions between these genealogies. So here is just for these 3 genealogies, you've got the 2 transition times, and then you can do that for the entire genome, basically. So this basically gives you this sort of in a very approximate transition matrix that you can then use to estimate the demographic parameter. So this is a very brief overview of the method. So we basically used this method to estimate the demographic history using both SNPs and SMPs. So if you look at the panel on the left, that's for the genetic variants, and you can see at about 10 to the power of 5 generations ago, it's kind of picking out this kind of bottleneck event and expansion, which we already knew has happened in these 2 populations. However, if you look at the SMPs, that same interval in time, you see that there's a little bit more noise. It's not so consistent, and so we think perhaps SMPs are not really that useful that far for capturing events that happened so long ago. However, SMPs are capturing this very recent population expansion, right, that cannot really be detected using SNPs alone. And we did some additional testing and simulations to ensure that this is not simply an artifact of this higher rates of change for the methylation marker. So methylation seems to be capturing some recent evolutionary changes that cannot be detected using genetic variants alone. So the final set of results I want to highlight is our efforts to identify group structure using methylomes. So for this part, I worked closely with Andreas, who was a master's student in the group, and we were basically working with methylomes that were generated from 2 different clonal groups of Brachypodium distachyon. So this is a clonal grass species. So Samples within each clonal group have very high levels of genetic similarity. So you basically cannot really separate them using SNPs alone. So we had paired a methylome and phenotype data for these two clonal groups. So the first thing that we did was basically just took each pair of samples across across these two clonal groups and just compared the proportion of methylation sites that differ, and generated a distance matrix, and that's being represented as a tree here. So what you see is that regardless of whether you use SNPs or DMRs, you can see obviously the group 6, which is the blue, the ones labeled in blue, they're kind of separating out quite well. But interestingly, within group 1, there's a very clear separation between these two subgroups that, again, you could not identify using SNPs alone. And interestingly, these two subgroups, I don't know how easy it is for you to see here, but these two subgroups actually differ quite a bit in terms of various morphological traits, especially related to seed morphology, so things like seed width, seed yield, seed length, and a variety of others. And also, the samples from these two groups actually they seem to be occurring at slightly different elevation. So methylation, at least in this case, seems to be picking out real differences that cannot be detected using SNPs alone. So I'll just summarize the list of main findings. So we think that SNPs, or using individual sites, are better than studying DMRs for performing within population analysis, partly because they're less influenced by these regional variation. We also have some evidence to indicate that methylomes and genomes seem to be evolving in unique ways at a given local level, that is. We think that methylation can be quite useful for revealing very recent demographic events, whereas genomes are perhaps are better for revealing the older ones. So basically, they kind of complement each other. And finally, that methylomes are quite useful for uncovering group structure in clonal species. So with that, I would like to just thank the Engelhorn Foundation for funding my postdoctoral fellowship, as well as all the group members at the Tellier Lab at the Technical University of Munich. And thank you all for listening, and I'm happy to take questions.
We have time for a few questions.
Yeah.
Yeah, can you explain what's the difference between the SNP and DM differentiated methylation?
Sure, yeah. So the question of what's the difference between an SNP and a DMR. So an SNP is basically just an individual site, individual CG site, where some samples have them are methylated, other samples aren't, whereas a DMR would be sort of an entire region where, yeah, some samples have very high levels of methylation, others don't. Yeah.
Is your colleagues' deep learning method for population divergence detection publicly available?
Not yet. So it will happen at some point, yeah. So, I mean, part of that will be associated with this, which will hopefully in the next few weeks.
What's it called?
No name for it yet. We're still deciding. If you have some suggestions, I'm quite happy to hear.
We have time for another quick question or two, or naming suggestion.
Yeah, I guess. Oh, yeah.
All right, the methylation in CG works in a clock-like fashion, but in the CHH less so, right, because it's less heritable. But is there like a change in the mutation rate, in epimutation rates between these two contexts?
So I have to admit, I've mostly focused just on the CG for my work because that's the one where the evidence of the transgenerational inheritance is strongest. So I think there are some differences. So this one is at the rate of 10 to the power of 4, but I don't know off the top of my head what is it for CHH, sorry.
No, I was just wondering because they are maintained by different molecular mechanisms, right?
Yeah, exactly.
There's any estimate of the difference?
There definitely is. I just don't remember it right now, but there definitely is, yeah. I think it's definitely high, it's not low. It's an order of, you know, it's not close to like mutations or anything. It's very high, I just don't know the exact number. Yeah.
All right, thank you so much, Arun.
Thank you.
So hi, I'm Hannah Waterman. I'm a PhD candidate in Trevor Krabenhoff's lab, and today I'm going to be talking to you about DNA methylation and whole genome duplications within the catastomid fishes. We've talked a lot about DNA methylation today, so I'm not going to go over this too in-depthly, but I do want to point out that I'm talking about DNA methylation and more specifically CPG methylation. So, so when we look at DNA methylation in fish, we find that approximately the global methylation rate is around 70 to 80%. You can see this in these lovely figures that I pulled out from Metzger and Schultz 2018 paper on the 3-spine stickleback. So here we have 5 different chromosomes, which you can see the average methylation is around 75%, and we have these odd little divots. These little divots are actually called DMVs, or DNA methylated valleys. These are stretches of hypomethylated valleys that generally span 1 to 2 megabases, and they also contain crucial developmental gene clusters such as HOX and protocoherin. Protocoherin is kind of how you develop neural pathways, where HOX is actually how your body develops, so very important. So here we have multiple different organisms and the HOX clusters they have. They are colored by their gene and where they are in the body where they develop. Specifically today, we are looking at teleos, which you can see have duplicated HOX clusters. We also will be talking about whole genome duplications. And so if you imagine when a whole genome duplicates, you have twice as many genes as you had before, so evolution can act upon those. So you can have non-functionalization, where we have one gene remains and the other one becomes fractionated or deleted, or even just it becomes a pseudogene. We have neofunctionalization, and this is where one remains the same while the other take up a whole entire new function, or subfunctionalization, where they divide up the previous function, which you can think of this more as tissue-specific expression. So multiple different organisms can go through and have gone through whole genome duplications, such as fungus, plants, as well as animals from invertebrates to vertebrates. So generally speaking, after a whole genome duplication, you would see something that looks like this, where we have 2 different subgenomes and we have about similar gene expression and similar methylation going on. But through time and evolution, we can actually end up with something like this sometimes, where we have a subgenome with a higher gene expression and higher methylation, and a subgenome with lower expression and lower methylation. And this is often because relaxed selection is acting upon it. So again, we are talking about teleos today, and so all teleos have went through a teleos-specific 3R duplication event around 320 million years ago. And this duplication gave rise to things like Danio, our zebrafish, or the 3-spine stickleback. Today, we are going to be looking at a family that went through an additional duplication around 65 million years ago called the 4R Catastomidae specific duplication, and we're going to be looking at 5 different species inside of that family. I do want to point out that the unduplicated outgroup, if you look at Danio, has about 25 chromosomes, and then when we look at our additional duplicated family, we see that they have around 50. So if we plot this on a synteny plot, looks like something like this. So we have Myxocyprinus, and it's 50 chromosomes on one side, and then Danio, and it's 25 chromosomes on the other side. And there's this really gorgeous 2-to-1 ratio that we're seeing here. So if you look at the extremely dark red color, it is chromosome 7 and 8 in Myxocyprinus goes both to Danio chromosome 1. So if you imagine, this duplication can open up a lot of interesting questions, especially when we go back to those DMVs. Are they duplicated across this duplication, especially since we're seeing this amazing conservation? So our key questions here are, what are the patterns of these duplicated species, as well as do we see evidence of subgenome dominance? And if we can methylation help us understand the evolutionary process following this whole genome duplication? So we use this really great technology that I'm quite a fan of, mainly because it actually takes DNA and pulls it through a pore and gives off an electrical frequency. This electrical frequency can tell you base pairs, as you can see at the top. There's a cytosine next to a guanine with its electrical frequency below it. We also can get, for a free byproduct, methylation. Yay! So you can see here there's a methylated cytosine next to a guanine and its electrical frequency. So given this, I was able to take stuff that was already sequenced by our lab and some stuff I've already sequenced and create 7 species methylomes across multiple different tissues. So we have muscle, gill, and blood. Our coverage for our methylomes are around 20x coverage, with our lowest being about 17.5. So again, our duplicated for our duplicate Catastomidae family is in the red box. And then our unduplicated outgroups, such as Gyrinocylus and Danio, are also we created methylomes for. This is a very large plot, and I do not want to overwhelm you, so we're going to start really slow. So if you look at the very top corner, we are just zooming in on those two chromosomes. So we have chromosomes 7 and 8, and if you remember from the synteny plot, those go to Danio 1. So remember, 7 and 8 are duplicate chromosomes or homologous chromosomes. And these are on a sliding window plot of 5,000 CpGs. So we have chromosome location on the x-axis and methylation frequency on the y-axis. And we have both muscle and gill tissue for Myxocyprinus. I promise you gill is there, but it's just very similar to muscle, so it's kind of hiding behind it, but we'll see it more when we zoom in later. Okay, so now all 50 chromosomes. Here we go. We have 25 boxes of duplicated chromosomes, giving a total of 50 chromosomes. So the overall methylation for Myxocyprinus was about 83%, and it didn't actually vary very much across all 50 chromosomes, which we thought was very interesting. And more surprisingly, we see this absolutely gorgeous mirroring effect across our duplicated chromosomes. And if you haven't noticed by now, I hope you can see that there are some little divots, if you remember our DMVs. So I wanted to statistically pull those out. And I did that by taking all of the points from that previous massive graph and plotting them on a histogram. So methylation frequency is now our X, and frequency is now our Y, and we have gill and muscle tissue. So then we pulled out the 95.5 percentile, which is that beautiful little dotted line, and everything to the left of it became a DMV. I did do some filtering, so that way 2,000 CpGs had to be in the DMV to make sure I'm spanning a very large region of hypomethylation, because DMVs are known to be large. So going back to Myxocyprinus, what we found that we had 10 hypomethylated valleys, and these are the top 10 deepest and the longest. If you do notice, they are in 5 pairs of homologous chromosomes, and when we actually went in and annotated these, we found that they are protocoherent and Hox clusters, so extremely important, crucial genes to developing. So just so we have an idea of what these hypomethylated valleys look like when we zoom in, we are going to look at Hox AA specifically. So let's zoom in. This is a plot, so we're no longer looking at 5,000 CpGs. It's just 100, so much more zoomed in, finer scale. So we have chromosome 30 on top, and then chromosome 30 genes, and then chromosome 34 genes, and then chromosome 34 methylation. As you can see, we have absolutely gorgeous hypomethylated valleys. We have it encompassing the whole entire gene cluster, starting at 1 and going to 13. 14 was not in the ancestral duplication, so we don't have to worry about it. You can see there are some genes missing here, but in generally, it's a very conserved area with this great, gorgeous hypomethylated area with tissue-specific methylation going on inside. So naturally, we wanted to see, does this persist in the other 4 species? And guess what? It does. So here are, this is actually Hox cluster CA that you're seeing here across my 5 species. And you can see that they all have conserved hypomethylated values that are duplicated across this duplication. And so I wanted to just take a little closer look inside of these hypomethylated values just to see what's going on inside. And so, of course, we're going to start with Myxocyprinus because it's my favorite, I don't know. So here, again, we have the same kind of thing that we're looking at with the 100 CpG average sliding window. But we did see something slightly different, right? We have that really gorgeous conserved hypomethylated valley here, but it seems to have started from 4 and goes to 13, which is really odd. So Hox cluster CA1 is kind of off by itself in its own little valley, and then the methylation reverts to the higher methylation of where 2 and 3 would be, but they are no longer there. And then we have the conserved valley. This was very odd that we found it in Myxocyprinus and found it across the duplication. So naturally, my next question is it in the other 4 species? And it is. So again, we are seeing this pattern where we have a little tiny divot where Hox cluster CA1 is falling in. then a reversion back to the higher methylation, where 2 and 3, because they have fractionated away. And then we have these beautiful conserved hypomethylated valleys, again, where it holds 4 to 13, which we thought this was very odd, especially from the fact that some of our annotations didn't even pick up HOXCA1. And we hypothesized that this is actually due to because either the gene is getting lost and becoming a pseudogene or fractionating out, and where we're watching the methylation kind of reverting back to that higher methylation, but we're watching it separately across all 5 species occurring since this whole genome duplication. So let's just go over our findings one more time. So we found that the overall methylation average was around 82% for my 5 species, which is slightly higher than our unduplicated outgroups, which makes sense, right? They're bigger genomes and they have more transposable elements. We did not find any evidence of subgenome dominance in the catastomates, though there has been 65 million years since the duplication, plenty of time for a subgenome to evolve. Sorry. And we did find some really remarkable conserved methylation across duplicated chromosomes with specifically 5 homologous pairs of hypomethylated valleys that have persisted these 65 million years. And then finally, we were able to find this really great relationship between functional genomic regions and methylation, as well as the similar but separate decay of Hox cluster CA. With that, I would like to thank everyone, lab mates, of course, and funding from Schonberg, CAS, and GSA. And I also would love to send a huge shout out to the amazing woman that sent me her Danio Ririo Fast Fives, because we sequenced everything in-house except for Danio Ririo, so luckily she was very willing to share that with me, so I was able to create that methylome as well. Finally, I will take questions, but first a little selfish little plug for my poster. I will be giving a poster at 5:45 today. Come see me at I-13 if you want to learn about tissue-specific methylation in an invertebrate. Thank you.
We have time for a few questions.
Do you think that the divergence in methylation, the tissue specificity that evolved very early after the genome duplication, so that it then was inherited through the speciation event, and that's why you see similar patterns in all of the species?
Yes, so the only one that I can say consistently, so we do have Myxocyprinus. Don't know if I can, I'm not going to scroll through. But Myxocyprinus, which is an early divergent species, which had that tissue-specific, and then Razorback sucker or Xyrauchin, which also had the gill and muscle that had that really drastic change in between the two. And the Xyrauchin is a much later divergent species, so maybe, yeah, because we're seeing in a Myxo, which again was the earlier divergent and in a later divergent. Go for it.
I was wondering, did you, when you're looking at the valley, find any ones or like any genes in there that you thought were important that weren't developmental, or was it almost all Hox developmental sort of genes?
I mean, I will admit that there were about 85 DMVs per each species. I really just specifically have looked at the biggest and longest ones, which are my Hox and Protocahurin, and been really kind of getting into the nitty-gritty of that. I think that I should start looking at my other ones, just a matter of time. Any more questions?
Very quick one.
If anyone wants to know, HOXCA is actually has to do with the Welvin apparatus as well as swim bladder lobes, which is really interesting because our catastomates has a 2/3 chamber swim bladder from this duplication. So that's just a fun little fact. If you have a question, I don't know if you have time.
Very quick.
Very quick.
You picked gill, which is constantly interfacing the environment.
Yes.
And the biology. Do you think that you still have the development of limbs and like Hox genes? We're thinking early development, and now we're looking at adult fish and gills. Do you feel that there's like a way of picking other tissues that are less changing or less interfacing with the environment and seeing different effects?
I understand where you're coming from exactly, but you know, we are seeing this also in muscle and blood, which are not interfacing in the environment. But gill definitely does tend to have deeper valleys. So maybe from the fact that it is interfacing into the environment is why they have the more drastic methylation compared to like muscle and blood. All right, thank you so much.
Thank you, Anna. Hi, everyone. My name is Paula Escuer. I'm a postdoctoral researcher in University of Neuchâtel in Switzerland. Today I'm going to talk about exploring the epigenetic landscape across Lepidoptera and its impact on chromosomal rearrangements. So I'm going to start introducing our model system, which are Lepidoptera or butterflies and moths, one of the most beautiful and charismatic group of insects, no doubt, but also one of the most diverse, with more than 170,000 described species. So Lepidoptera have allocentric chromosomes, which is a very particular characteristic. Instead of one centromere, which is in purple color, they present multiple centromere-like structures, and that have important consequences on their genome, as this allocentricity facilitates fusions and fissions of chromosomes. For example, in a case of a monocentric chromosome, if there is a fission during cell division, that will definitely have a strong impact on their fitness. However, in an allocentric chromosome, this impact can be highly reduced due to this multiple centromere structure. Also, this rearrangement could promote speciation through processes like hybrid dysfunction or recombination suppression. So this definitely had an impact in Lepidoptera because they present a large variability in karyotype. So in this plot, you can see the number of chromosomes in the y-axis and the number of species in the x-axis. So we can see that most of the species present the ancestral karyotype, which is 31 chromosomes, but we can find some of them that have 50, 100, or even 200. So some genera present a huge interspecific karyotype diversity, like is in the case of Erebia butterflies, that can have from 7 to 52 chromosomes. And we are very interested to know which processes are involved in this karyotype variability. So repetitive elements may play a role in that, as they can cause rearrangement via ectopic recombination. Here we have, for example, a transposon, and ectopic recombination is when there is recombination between two similar sequences that are located in two separate regions of the chromosome, and that can have an output like rearrangement, like inversions or fusions, etc. Interestingly, in the case of Erebia butterflies, which are the ones in this phylogeny, it has been found phylogenetic association between chromosome number and some particular repeat families, like is in the case of ribosomal DNA. However, some of the most abundant repeats in Erebia, which, for example, lines, they show no association with karyotype changes, and that could be because methylation have a role in silencing these transposomal elements to avoid this process of ectopic recombination. So as we talk in our recent published review about 3D genomics and speciation, we found links between epigenetic regulation and change in the 3D genome structure that could promote speciation at the long term. And also, there is a relationship between the genomic rearrangement and epigenetics. So definitely epigenetics and more concretely methylation is something that we wanted to explore to answer our main question. And our main question could be disclosed in 3 more concrete or specific questions. Like first of all, how variable is the methylation landscape across Lepidoptera? Also, does methylation play a role silencing or protecting particular regions from rearrangements? And the third one, does the Lepidoptera genera with more karyotype present a particular methylation pattern? So for that, we proceed with fieldwork last summer, 2023, across all Switzerland. It was a magnificent experience. And once we selected our data set, we proceed with the DNA extractions of the thorax part of the butterfly. Then we apply the bisulfite treatment, and we sequence whole genome bisulfite sequencing from 65 individuals from 35 different species. And here I want to thank the Project Psyche, which is a collaborative project between several institutions in Europe that aims to sequence the 10,000 Lepidoptera genomes of Europe. And thanks to that, this project has been done. So again, thanks Psyche. And again, saying thanks Psyche because some of the new genomes that we obtained are sequenced with the latest PacBio technology that also includes 5MC methylation data. So we had another question that was, does this PacBio data is coherent with the bisulfites? So we want to compare them now that we have 2 data sets in Lepidoptera. So now I'm going to present the results. I have to warn you, these are very preliminary results that I made these past 2 weeks or 3. And first, here we have the person correlation of the CPG sites in 2 different species, Lysandra corydon and Erevia oeme. And we use one female sequenced with bisulfite, a male with bisulfite, and a female sequenced with PacBio. And here we have the values of the slope. We see a high concordance between the 3 datasets of Lysandra corydon. And in the case of oeme, we also find concordance, especially more between the male of bisulfite and the female PacBio. Something seems wrong with the female, but we still need to do more work with that and also take on account other parameters like coverage or more things. And here I present you the global level C methylation, percentage of methylated Cs, separated by the different taxonomic families. The number that you see in the box is the sample size, and we separated by males and females. So we observe general levels of methylation of 1.4 to 2.7%. We found also, we see differences between males and females, but we don't see a clear pattern of females being higher or males higher. It seems more family specific, this variation. And also we found higher levels of methylation in the family of nymphalids. Another super interesting cool result that we found is that we saw a phylogenetic association between repeat content and methylation levels. And that could be because this increase of the repetitive elements could also generate an increase of the methylation as a regulatory response to protect from these ectopic recombination processes. Another thing that catch our attention is that Erevia species, the ones that are in the circle, they present also one of the highest levels of methylation of all the species that we analyze. And if you remember from the introduction, this family has high karyotype variability, and also it has been published recently that the speciation rates are higher with increased chromosomal change in this family. So definitely something that we will look forward. However, in this project, we want to focus on the main picture in the microevolutionary role of methylation. And for that, first we annotate already all the repetitive landscape of these 35 chromosome level genomes. Here I show you just 2 of the 35 plots, and we can see that there is a lot of differences between the repetitive landscape in different species of butterflies. We proceed with synteny analysis. This is very cool, but probably you don't see much. It's not very informative, but we can see that some families are super rearranged and other more conserved. And the next step will be to identify breakpoints and the methylation patterns in these breakpoints. And just to finish with some take-home messages and trying to answer my questions. First of all, we found low methylation levels and differences between taxonomic families and males and females. We found significant correlation between the proportion of repeats and methylated cytosines. We found also some clades with higher karyotype variability that present also high methylation levels. And we found significant correlation but intermediate concordance between both methylation data sets. And finally, I want to thank all my colleagues from the Biodiversity Genomics and the Parasitology Lab of the University of Neuchâtel, and to all of you for your attention. Thank you very much. The one, the one that was expanded, you mean? Ah, the ribosomal DNA. You, no? Ah, yeah. Yeah, I think are rolling cycles. I'm not super expert in repeat families, but there seems to be high variability between the species. And also, if you see the lower plot is the Erebia OM, and we can see that a lot of the repeats are very new. And in the other, I think it's a Pierit, I don't remember now, but seems that have more old elements, more or less. But I'm trying to focus more on the methylation part in my project. Yes?
I'm curious, do you think these between species methylome variation is also mirrored in earlier life stages, like when they're caterpillar or before they form a cocoon? Or do you think that you've seen similar types of patterning between species?
Sure that they should be differences between other stages because they have been done some methylation studies in butterflies and they found differences between larval stage, so that's for sure. And also I'm focusing now in the adult stage, but yes, I think, yeah.
I have a question.
Mm-hmm.
What do you expect to find at the chromosomal breakpoints in terms of methylation differences?
I will expect that they have to be high methylated, I guess, because these are points that are easy to break or to have some kind of rearrangement. I guess that methylation has to try to protect these areas because we don't know, but probably even if in allocentric chromosomes the impact is not as high that in monocentric, probably they have a slightly deleterious effect. So I guess that methylation has to protect, but still we don't know why some clades have so many rearrangement and others are so conserved. So that this will be something very interesting to resolve in the future.
Time for one more quick question.
So in Lepidoptera, a lot of the methylation occurs within gene bodies. Did you find that?
I still didn't have time to look at that. I have a lot of plans to do, but I show you just some things that I had time to do to show something at here. But yeah, that would be a very cool thing to do also.
We have time for maybe one more quick question. Nope. All right, thank you, Paula.
Thank you.
Okay, thank you very much to having me here as the last speaker. I hope you guys are still waking up. So I'm Juntao Hu from Fudan University. It's glad to be back in Montreal because I did my PhD with Rowan over there back to 2014. And after I left his lab, I'm still doing epigenetics. And here are some of my recent work and ongoing work that I hope will be feeling interesting. So as a lot of guys talking about today, the animals will respond to a lot of stressors or environmental changes in two different ways. One is the plasticity as a short-term mechanism. The other one is the evolution as a long-term strategy for animals. So my interest focused on the epigenetic mechanism underlying the plasticity and evolution in the vertebrates specifically. So I've been working with a lot of different animals, from sticklebacks, to guppies, to the mosquito fish, to anole lizards, and from the star tunicates. A lot of the different systems are built on the work in Rowan's lab, but also expanding my work into different systems. I'm specifically interested in the epigenetic change underlying plasticity and evolution, because as you can see here, it's a layer between the genome and transcriptome, and finally you can see from the phenotypes. So actually it's a good connection to the genetic variation to the phenotypic variation we observe in nature. So a lot of guys today are very familiar with epigenetics. I'm not going to talk about the concept of epigenetics, But in my work, I'm specifically interested in the DNA methylation and also a different epigenetic modification called small RNAs and specifically microRNAs. So those are the two epigenetic mechanisms that will activate or suppress gene expressions and also have influence on the phenotypes. So for my talks, I'm gonna have it divided into two different topics. One is about plasticity, the other one is about evolution. So for the plastic side, I'm interested about how extreme temperatures and freshwater salinizations will have influence on the plastic response of the animals and how animals will use epigenetic variation to respond to those two different kinds of environmental stressors. So for the extreme temperatures, we did experiment on the mosquito fish, and the experimental design was pretty simple. We just expose the fish to the extreme high or low temperature. We call it critical temperature maximum or critical temperature minimum, where those two temperatures, when the fish reach the two temperatures, they will basically unable to remain their balance in the water. So we collect those, or collect fish at the depths immediately after the fish reach those two different temperatures and measure their methylation variation, gene expression variation, splicing variation, and also microRNA expression variation. So basically, we found out that at these two different temperatures, a lot of genes that are showing variation at those different 4 levels are not overlapping, as you can see from the Venn diagrams. And also, it's a little bit unexpected that we did not find any differential expressed microRNAs, suggesting that the different epigenetic mechanism do not respond to the same environmental stressors at the same time. They may have some different time lags between those two different epigenetic mechanisms. So from the freshwater salinization side, one of my students already showed those results in the morning. So basically, we collect fish from 2 different environments. One of the population had previous history of salt exposure, the other one did not have. And we stress those 2 different populations to a project salinization levels, where we expect you to have these salinization levels by the end of the century in some part of the world. And basically, we found out that at the 3 different time points, at 1 hour, 24 hours, and 48 hours, a lot of genes that are showing differences between the the salinity stress and control salinity levels in these 2 populations are parallel. And for those parallel genes, when we analyze in their nucleotide diversities, for the parallel genes that are showing parallel response across different time points, they have higher nucleotide diversity levels than the other genes, suggesting those genes might provide higher evolutionary potentials for the freshwater populations to respond to future salinizations. And the other results we had is that for those parallel genes that are showing the same direction of expression change across time points, they are regulated largely by different microRNAs. So we are thinking that probably the microRNA are important non-coding modulators for the freshwater populations to respond to future salinization. Okay, so for the other side of the story about evolution, we are interested in 2 different specific questions. So the first one is sitting on the parallel evolution side. I'm pretty sure a lot of you guys are familiar with parallel evolution. It basically describes a phenomenon where the similar environments can predict similar phenotypes between populations from the common ancestor. It has been shown in a lot of different systems, from the cichlid fish to the well-known three-spine stickleback. A lot of studies have analyzed parallelism at genetic and transcriptonic levels, but we were trying to analyze the parallel evolution at the epigenetic levels. And also, besides epigenetic variation, a lot of the molecular mechanisms can be underlying the parallel adaptation. So we were also interested in how those different mechanisms interplay during parallel adaptation. So when we analyze the level of parallelism at the epigenetic level, to be specific, at the demethylation levels, when we comparing 3 different pairs of marine and freshwater ecotypes of the three-strand stickleback, we find a non-significant tendency for epigenetic variation to be parallel. When you're looking at those dot points or scatter plots, as you can see, a lot of the methylation do show parallel change between different pairs of ecotypes, but there are still a lot of methylated sites showing non-parallel change between pairs of ecotypes. So we were thinking that unlike the parallel evolution at other molecular levels, probably there's no tendency for epigenetic variation to be parallel. And also because one of the studies or one of the pairs of the ecotypes we used in their studies, they also have a reciprocal transplantation experimental design. So we took advantage of that reciprocal transplant design and analyzed how plasticity will facilitates or hinders evolution. So there are the results showing that regardless of the evolution directions, whether we analyze in the freshwater marine, the marine population entering freshwater environments or the freshwater ecotypes readapted to the ancestral marine environments, the majority of the methylation or the plasticity actually showing the opposite direction to the divergence between the marine and freshwater ecotypes in their adapted environments, suggesting a lot of the plasticity were maladaptive and they will hinder evolution. And also, we also took advantage of the reciprocal transplant design and analyzed whether the proportion of plasticity versus evolved change will be different when the direction of adaptation changes. So we analyzed 2 directions of adaptation. One is called freshwater adaptation, which mimics the real evolution directions where the marine ecotypes will enter freshwater environments and diverge into freshwater ecotypes. The other one called marine adaptation, which is just the freshwater ecotypes readapted to the marine environments. And our results showing that in the freshwater adaptation, a lot of the methylation change is due to evolved change, and as opposite or in contrast, when the freshwater population meet their ancestral marine environments, a lot of the methylation change in the freshwater ecotypes are due to plastic change. So those results suggesting plasticity will be likely to be a memory for the derived population to remember their ancestral environments. So here's the other story. We try to analyze the role of epigenetic variation in evolution. So here we compare the relative roles of genetic versus epigenetic variation in adaptation. But here we are using an invertebrate called star tunicates. It's a kind of invasive species in the marine environments, and worldwide it has been distributed into different environments. So it's a pretty good system that can allow us to test the association between epigenetic or genetic variation and environments. So we using the same populations I just showed you, we analyze the association between genetic variation and environmental variables, and also epigenetic variation and environmental variables. Regardless of whether we're analyzing all loci or the neutral loci or adaptive loci in genetic and epigenetic levels, they are showing quite different structures between epigenetic structures and the genetic structures, suggesting probably genetic variation and epigenetic variation are independent from each other and allow the animals to respond to the environments in an independent way. And also, we confirm our results by analyzing the adaptive genetic loci and adaptive epigenetic loci at functional level. We observed that a lot of the genes associated with either adaptive genetic or adaptive epigenetic loci are quite independent or largely non-overlapping. So again, confirming our hypothesis that genetic and epigenetic variation might be independent from each other in the evolution. So to summarize my stories, so when we analyze the epigenetic basis of plasticity, we see that epigenetic change can occur very rapidly because our experiment only lasting for hours to days, compared to the genetic change, which might take generations. And also we found non-concordant epigenetic and transcriptional responses to the acute small stress, suggesting that different molecular levels might play different roles. And also we observed that the parallel plastic response may be regulated by different epigenetic modulators, suggesting that especially for microRNAs, which can where the one genes can be regulated by different microRNAs, and so the microRNAs might provide higher potential for the animals to respond to the environmental stressors. And at the other side of the epigenetic basis, which is the epigenetic basis of evolution, we found out that although there's a lot of tendency for gene expressions to show parallel evolution, but we found no tendency for methylation to be parallel. And also epigenetic plasticity may be adaptive during the early stage or the early stage of colonization of novel environments, especially in the three-strand stickleback system, and also our results suggesting probably independent roles between epigenetic and genetic variation in adaptation. So with that, that concludes my talk today, and I'm happy to take questions. Thank you. What do you mean by there's no point on the axis?
Like you have like the 4 cones going out in each quadrant, but you don't have any data points that fall like right in between parallel and non-parallel. So is there like a way that the metric is calculated?
Oh, okay. So for each of the 3 scatter plots, like take panel B, for example. So those dots are one of the DMCs between marine and freshwater in one pair of the stickleback. And also when we identify a DMC between marine and freshwater ecotypes, we use a pairwise comparison between two different pairs of the stickleback. And when they showing the same direction of methylation change, we think that will be a parallel change. So as you can see in the first and third quadrant, they are showing the same direction of methylation change. That's why we'll call it a parallel. And in the opposite direction, we call it non-parallel. And yeah.
So if it fell on the axis, it wouldn't be a DMC, either, is it not?
Actually, we didn't find any DMCs falling on the axis, because if there's no methylation change between the marine and the freshwater ecotype, it won't be called as a differential methylated cytosine. Thank you.
I'm not an epigeneticist. I like the fact that we define epigenetics as non-genetic traits that are heritable. What I struggled a little bit with is you talked about epigenetic change over hours to days. You're just saying methylation change, right?
A lot of the studies here are about methylation change.
So it's not necessarily epigenetics.
If you consider...
If you define epigenetic as something that is heritable across multiple generations?
No, none of the studies here are necessarily about heritable epigenetic variation. So if you are talking about plasticity, we are doing all experiments within generations, so there's no heritability. So about the evolution side, we are comparing pretty much population epigenomics. So I can imagine some of the methylation here won't be heritable across generations if the environmental cues were removed. So we did have a study. I had a study with Rowan back to 2021. We did analyze methylation change across generations. In that study, we did not have any environmental change, but we do observe a significant amount of methylation change, so our methylation levels stay stable across generations. But again, I won't call that heritable because you need to analyze whether genetic variation will play a role in controlling methylation levels. But we also analyzed that. In that study, we only found about 30% of the methylation levels were controlled by genetic variation. So a lot of the methylation variation can be heritable and are autonomous from genetic variation.
So in the last panel there, the genetic structure versus epigenetic structure are not the same.
Mm-hmm.
In the epigenetic structure, is there like ecological convergence? Like are populations that might be otherwise less related than a neighbor, but then enter into similar environments? Do those environments force convergence of epigenetic structure?
I wouldn't say force, but epigenetic variation is more sensitive to environmental change than genetic variation. We did analyze the environmental distance, or AIBE. Here, as you can see, there's significant correlation between epigenetic variation distance and environmental distance. So yes, to your question, if the environments between two populations are similar, they might have similar epigenetic levels. Yep.
Thank you, Juntao.
Thank you.